to Ecology is Hot. Today we're talking about predator-prey relationships. Perhaps the hottest relationships of them all. Okay, so there are different types of predators. There are true predators, which are like big cats and wolves, where they are removing individuals from the population of prey. So they kill it while it's alive. Then we have parasitoids, which are like wasps and some flies, who lay their eggs on or inside other organisms. And then the eggs hatch, and they eat whatever organism they were put inside. So parasitoids also kill the host. Then you have parasites, which usually don't kill the host, because they rely on the host to live. And then you have herbivores, who don't actually kill plants, but they will clip certain parts, like perhaps the leaves. Predators are characterized based on what they eat, so they could be herbivores, carnivores, omnivores, nectivores, eating nectar. They could be sanguinivores, eating blood, like vampire bats and such. And then they can be characterized based on their effect on prey organisms, which would be your true predators, parasitoids, both of which kill their quote-unquote host, and then parasitoids and herbivores, which leave their host not unharmed, but undead. So predation can be a realized niche boundary determining factor, and it's a density-dependent factor, but it's actually rarely true that predators control prey populations. But it is density dependent. Okay. So some examples of true predators are big cats, wolves, things like that. Parasitoids are typically wasps, and usually the hosts are larvae or caterpillars. So these wasps have an omnipositor, which allows them to put their eggs inside the caterpillar. Some wasps can even tell if a certain caterpillar or host has already been infected. And some parasitoids, called secondary parasitoids, only inject their eggs into other parasitoids. And then there are tertiary parasitoids who only lay their eggs inside secondary parasitoids. Crazy, right? It's kind of complex. Alright, parasites often have a primary and a secondary host. So if you look at this diagram, you can see that it starts as a little cell and it goes in something else, and then it turns into a mosquito, and the mosquito eats your blood, and yeah, all that. Okay, we have omnivores, and it was seen that where wildebeests were grazing, there was actually more biomass of plants. The biomass increased where the wildebeests were grazing, but it decreased on ungrazed areas. So wildebeests can actually create more plants while they eat them. And they can do this because they're uncovering layers of vegetation, so exposing them to sunlight. They can help distribute the seeds. They, can deposit fertilizer. Most importantly, wildebeest saliva actually has an enzyme in it that can stimulate plant growth. Now, that's obviously a good thing for the plants and for the wildebeest. So herbivores are the only category where the prey can benefit like this. So the prey is benefiting because there's going to be more biomass. They essentially help the plant grow. So that is not true in true predators, where they get eaten. Okay. So, a practical interest in predator-prey relationships began in the late 1800s because of agricultural pests, obviously. It continued as a theoretical interest stimulated by the presence of population cycles. So, we noticed through the Hudson's Bay Company data that the effects on predators and prey, well, they affect each other's numbers So there's this presence of a population cycle. So when we attempted to stimulate cycles in the laboratory, we found that artificial conditions led to better understanding of the roles of immigration and environmental heterogeneity. So by conducting these laboratory experiments, we saw that it was important to be able to have other organisms come into the experiment, the area, in order for the actual cycling that happens in the environment to happen. And there have to be places to hide, but the right amount of places to hide so that the predators can find them, but enough can hide that the species can still survive. Yeah, okay. So from an evolutionary perspective, predators are viewed as discrete, measurable events. Predation is viewed as small, measurable events such as pursuit, search, and handling items. So the time spent in the events can be measured, and alternative strategies could be used by individuals, and they can now be compared for effectiveness. So this led to the formulation of optimal foraging theory, which is abbreviated OFT. OFT. I feel like we'll talk about that later. Yeah, awesome. Okay. Well, that takes us to the Hudson's Bay Company, and a couple important things to remember about that is it was a very large corporation in North America, and it's given to us by England, and it controlled the trapping trade. But what was most important was it conducted this experiment, so to speak. So they have all this data from the furs that came in, because the trappers would bring their furs into the trading post, and they were paid according to the species and the quantity of the pelts. So they wrote all this stuff down. So the company kept really good numbers and species numbers, and some of the best numbers of animals out in the environment of anyone. So they have really good data, and it lasts for a really long time. That's the difference. So they came up with this cycling where there's like 10-year cycles. It's common to have 10-year cycles between lynx and snowshoe hare, which is the lynx's favorite food. Around here, it's more like a 4-year cycle since it's warmer. That has to do with food availability, climate cycles, gestation periods, and reproduction times of cycles. When there are fewer predators, the prey population rises. So when one is up, the other is down. So if there's no prey to eat, the predator levels will go down, and then since the predator levels went down, the prey goes up, so on and so forth. So they could infer the population levels based on the furs from the Hudson Bay Company, who took this data for such a large amount of years. So, okay, so then this experiment with, I think it was bacteria again. So we see these graphs that when you couldn't hide and no new organisms could come into the tank, essentially, both would go extinct. Then, when you let the prey hide, the predators became extinct because the prey could hide. And then, when you let individuals come in, immigrate, and there are places to hide, then you can reproduce that cycling in the lab. So this lab experiment showed that it's important to have not only hiding places and not only immigration, but both immigration and hiding places in order to reproduce this cycling of predator-prey populations. Okay, then Huffaker had this experiment with oranges and rubber balls to represent environmental complexity. He covered them with wax paper, except for the top, so that he could see where the mites were. Okay, so this also confirmed that you need environmental complexity to get the oscillations in laboratory populations. Excellent. Yeah, so that's what he did. Okay, so that leaves us to the Lotka-Volterra equation for predator-prey or parasite-host population growth. So on the left, we have the rate of prey or host population change. So the rate of change of the population. And then that's on the left. And then on the right, you have the host per capita rate of increase times the exponential rate of increase by host population minus the predation rate times the number of prey or hosts killed by the predator or parasite. So essentially, you have the rate of predator or parasite population change equals C, the number of calories in a prey item by how many are used by the predator for reproduction, times the rate at which prey are converted to predator offspring, so how many offspring are predators having as a result of eating that prey, times the number of prey, times the number of predators, minus the predator death rate, so the mortality rate of the predators, how many are dying, times the number of predator deaths. And that gives you the rate of predator or parasite population, whereas the first equation gave you the rate of prey population change. Okay, so based on this provides us with a mathematical basis for the cycling that we see. And when you look at the cycling, when you take time out of the equation and you graph prey numbers versus predator numbers, you get a circle like so. So this makes sense because as we see here, as the number of prey goes to the right, that means the number of prey goes up. So if you start in the bottom left, as the number of prey increases, well, as the number of predators is decreasing, the number of prey is increasing. And then in the bottom right, the prey keeps increasing, so the predators can then increase. And now, since the predators are increased, the prey heads to the left, which means it decreases. And then since the prey is decreasing, the predators have to decrease too, which allows the prey to increase once again. Which makes sense if you take a minute and think about it. So that is where your predator-prey oscillations come from. Right. Now, the effect of the density of prey population on how quickly the prey are eaten. So, this describes how predators behave, mainly based on how many prey there are. Okay, so type 1, as we see here, as the density of prey increases, the rate at which they eat it increases too. So if there's more and more, they'll eat more and more and more. They never get full. They just keep eating and eating and eating everything in sight. Eventually it'll probably level off, but that's off our graph. Type 2 is a classic pattern where when prey density is low, consumption rate is high, because it immediately consumes whatever is available. But it levels off due to handling/processing time as prey density increases. So you can only eat so much so fast. That's the important part to remember. Type 3 has a slow start, and then it eats exponentially, and then it levels off. So maybe prey is hard to find, maybe it consumes it seasonally, maybe it's an ambush, maybe it has to develop a search image. You know, any of these things could be the result of that slow rate in the beginning. Okay, so that's about all my time for this video. So we'll have to pick it up next time. Thanks for listening. Hope I'm not boring. And good luck on your test.